The small intestine. The small intestine includes the duodenum, which is part of the upper GI tract, the jejunum, and the ileum, which chemically digest chyme and absorb nutrients, water, and electrolytes. The small intestine, which is also known as small bowel, is a long tube that extends from the pylorus of the stomach to the cecum of the large intestine, which finishes chemical digestion and is responsible for absorbing most of the nutrients from our food. Generally, about 9 to 10 liters of ingested food, water, and digestive secretions enter the small intestine daily. Ingested nutrients spend at least 12 hours inside your small intestine. On the right is the key functions of the small intestine, including mixing liver and pancreatic juices, absorbing nutrients, continuing digestion of carbs, proteins, and also fat breakdown. It also produces hormones to regulate the GI system and also produces immune cells in large numbers. The nutrient digestion and absorption occurs inside the brush border. The histology of the small intestine. Inside the small intestine, there are tons of villi, or small finger-like projections of simple columnar epithelium and lamina propria of the mucosa in the small intestine. The villi are the largest and most numerous inside the jejunum, where a majority of absorption takes place. Each villus contains an arteriole, which is a blood capillary network, a venule, and also a lacteal, shown here with the green vessel, the red vessel, and the blue vessel. A lacteal is a lymphatic capillary within the villus that is responsible for absorbing lipids and vitamins, mainly lipid-soluble vitamin. Then when you zoom in closer, we have the microvilli, which are extensions of the plasma membrane of the columnar epithelial cells lining our small intestine, and they form what we call the brush border cells. Brush border also contains enzymes, which are enzymes embedded inside the brush border layer and complete the chemical digestion. Within the small intestine, we have the intestinal glands called the intestinal crypts, which are invaginations of the mucosa, and they secrete some of these intestinal juices. It can increase the small intestine's surface area by over 600-fold, and this is why having a lot of these hair cells are really helpful in the absorption process. There are several unique epithelial cells inside the small intestine. Absorptive cells are extensive microvilli. Their function is to produce the enzymes like the brush border. The globate cells within the simple columnar epithelium produces mucus, which lubricates and protects the intestinal lining. The number of globate cells increases as you go from the duodenum towards the ileum. So the number increases. Then we also have the granular cells, which provide immune-based protection. These are oftentimes mixed with the Paneth cells, which are located at the base of the intestinal crypts. They secrete stuff like lysozyme and other antimicrobial agents to help with the innate immune system. And last but not the least is also the enteroendocrine cells, which produce hormones such as cholecystokinin, like CCK, and secretin, which controls how much acid is produced inside the stomach. Here's a cross-section showing you the Peyer's patch, a layer of immune cells inside the small intestine. And its main function is for immune surveillance. Motility of the small intestine. Smooth muscle contraction. Smooth muscle of the muscularis of the small intestine mixes chyme with the accessory gland secretions. It moves the chyme against the brush border, propels the contents through the small intestine to the large intestine, and facilitates in the chemical breakdown and absorption. That segmentation that we talked about is a back and forward motion that mixes the chyme with all these juices, and the peristalsis propels it down toward the GI lumen. This area contains a lot of mucus, electrolytes, and water. Also, disaccharidases, peptidases, nucleases are also attached to the microvilli. They help with the breakdown of all the macros. Key hormones like cholecystokinin, gastric inhibitory peptide, and secretin are released into the bloodstream to help regulate the amount of acid being produced in the stomach. And last but not the least is that the accessory organs, like the pancreas, the liver, and the gallbladder, they provide bile salts for lipid breakdown and help with the enzymatic reactions for chemical breakdown of the macros. Lower intestine, or the large bowel, is a wide tube located within the abdominal pelvic cavity and absorbs water, electrolytes, vitamin B, and vitamin K. And any other remaining undigested materials get compacted into the feces. The gross anatomy is that it's about 2.5 inches diameter and about 5 feet long. It has 3 main areas. It contains the cecum, the colon, and the rectum. The key functions are it absorbs the majority of the water. It also has secretions of protective mucus and aids in the movement of non-digested food. This is also the site where most bacterial colonization occurs for specific nutrient digestion. This is also an area where immune cells are produced. Appendicitis is an inflammation of the appendix, usually due to some sort of obstruction by fecal matter. As the appendix becomes inflamed and swells, It compromises the blood supply and provides a site for proliferation of bacteria and inflammation. If untreated, in some serious cases, the appendix may burst, spilling its contents into the peritoneum, leading into further infections like peritonitis. In the early stages of acute appendicitis, pain can be felt in regions of the abdomen. Usually, the pain can be sharp and localized in the lower right quadrant of the pelvic cavity. And some of the symptoms include nausea, abdominal tenderness, elevated leukocyte count, and the main approach is to surgically remove the appendix. The large intestine, the colon, is partitioned into the ascending colon, transverse colon, descending colon, and sigmoid colon. The ascending colon originates from the ileocecal valve and extends superiorly, makes a 90-degree turn called the right colic hepatic flexure. On the other side, it also makes a 90-degree inferior turn called the left colic splenic flexure, and it makes a frame. There are specialized structures in the large intestine called the tani coli. These tani coli are these longitudinal bundles of smooth muscle that bunch up the large intestine into these sacs called the haustra, these pouches. The histology is composed of mostly simple columnar epithelium with numerous globule cells. They're smooth, but they do not contain villi. There are many lymphatic nodules that occupy the lamina propria of the large intestine. The muscularis layer contains 2 layers of smooth muscle, but the outer layer, longitudinal layer, is discontinuous and then eventually create into the tani coli. More than 90% of the chyme entering the cecum each day is resorbed. Let's talk about some of the key reflexes. The defecation reflex involves relaxation of both internal and external anal sphincters. Defecation reflex is initiated by the filling of the rectum, which results in increased nerve signals because of pressure, and they're sent along the parasympathetic motor neurons of the sigmoid colon. The rectum also has nerve signal that fire, causes the relaxation of the involuntary anal sphincter or the internal anal sphincter. So you can see that involuntary control is when the stool in the rectum is causing pressure to build up, so the parasympathetic begins to cause it to relax. This internal smooth muscle begins to relax. What's keeping the fecal matter in is the external anal sphincter. This is what's the bearing down. Eventually, when you're sitting on a toilet, this will then contract and allow pushing of the fecal matter. So it does involve the parasympathetic pathway to trigger. Motility and secretions in the large intestine. The peristaltic movement, the haustra churning, the mass movements of the tani coli propel the material through the GI tract. These key motility motions are caused by the enterocolic and duodenocolic reflexes, and this is triggered by the rectal wall being stretched or distended. Then the brain begins to initiate that defecation reflex. So the defecation reflex is initiated by the filling of the rectum, which results in increased nerve signal being transmitted along the parasympathetic motor neurons of the sigmoid colon and rectum. Predominantly, the secretions in the large intestine have very little with digestive activity. At this point, most of the stuff that's in there is going to become fecal matter. So feces then leaves the GI tract, mostly composed of water, mucus, undigested fiber, food parts, microorganisms, and also broken off epithelial cells. Accessory organs. Accessory organs, digestive organ tracts, include the liver, gallbladder, and pancreas, which release secretions into the duodenum. This is a setup that allows the secretion of the juices. The biliary apparatus is a network of ducts, including the left and right hepatic ducts, which then merge to form a single common hepatic duct. And the union of the cystic duct from the gallbladder and the common hepatic duct forms what we call the common bile duct. And this common bile duct will then eventually drain in to the duodenum of the small intestine. So you can see that all these ducts, the cystic duct, hepatic ducts, they kind of convert and intersect together, allow the juices to mix together. Over here on the bottom is the main pancreatic duct. And this is where the majority of the pancreatic juices come in to join. And there is a slight swelling over here where the juices will exit into the small intestine. So these accessory organs have a wide range of functions, including detoxify chemicals that are brought to the body, metabolize nutrients into other forms for storage, and also store bile to help emulsify fat for later usage in the gallbladder. Alright, let's start with the liver. The liver is an accessory digestive organ that produces bile. The liver is the largest internal organ that's located in the upper right quadrant of the abdominal cavity. It's composed of 4 lobes supported by 2 ligaments. We have the falciform ligament, and we also have the round ligament. The histology of the liver is quite unique. The liver's connective tissue capsule branches throughout the organ to form the septas, and they're partitioned into these hexagonal structures called hepatic lobule. Within these hepatic lobules contain hepatocytes, which are liver cells, and they contain the portal triad, which is composed of the bile duct, hepatic artery, and also the hepatic portal vein. So let's talk a little bit about how the fluid flows inside the liver lobule. The hepatic artery carries oxygenated blood to the liver. The hepatic portal vein carries a nutrient-rich but oxygen-poor blood from the capillary beds of the GI tract, the spleen, and the pancreas to the liver. The hepatic artery and the hepatic portal vein branch extensively into these smaller vessels under the microscopic branches of those portal triads. So the nutrients are absorbed from those regions of drainage and they enter the hepatocytes. Oxygen is delivered to the hepatocytes for cellular respiration. And there are also some smaller cells called stellate cells that engage in phagocytosis and cleaning up any kind of microbes or dead cells in the area. 75% of the blood enters through the portal vein. Only 25% of the blood enters through the hepatic artery. It's important because the liver does get the first dibs of all the nutrient blood. So as you can see in the image, that the hepatic portal vein brings blood flow from the stomach, the spleen, the pancreas, and also the mesenteric system all the way up before it goes back up toward the heart and being used to the body for getting oxygen. Bile is a yellow, greenish alkaline fluid composed mostly of water, bicarbonate, bile salts, bile pigments like bilirubin, cholesterol, mucin, and is produced by hepatocytes at a rate of about 0.5 to 1 liter per day. Biles are released from the hepatocytes into the bile canaliculi that transfer the bile into the bile ducts of the portal triads. Remember, bile flows in the opposite direction of the flow of blood. So the bile flows into the increasingly larger bile ducts until it reaches the left or right hepatic duct. So the key functions of bile include neutralizing acidic chyme with the bicarbonate inside it, emulsification of lipids by the bile salts and the lectin, elimination of the bilirubin, the waste of the red blood cells or erythrocyte destruction, and bile does not contain digestive enzymes for the chemical breakdown of any nutrients. It just makes it easier to assist in the breakdown of fat-soluble vitamins and fat-based compounds. So you can see on the right-hand side, we have a soapy layer. The bile salts and the phosphatidylcholine, or lectin, creates the soap layer. So the colipase and lipase can have better access into the fat droplet and then make it into smaller micelle formation. Makes it from bigger fat droplet to smaller fat droplets. The pancreas has both endocrine and exocrine functions. Endocrine functions of the pancreas produce and secrete hormones such as insulin and glucagon. The exocrine cells of the pancreas produce pancreatic juices that assist in chemical digestion. As a result, some of these disorders of the pancreatic ducts or pancreas have serious and even fatal types of effects on the body. The pancreas is about 5 to 6 inches, has a long head region, body and tail region. And the pancreatic duct is this big duct that combines with the bile duct to provide the juices into the small intestine. Its main function here is to produce the water secretions that's rich in bicarbonate and also plethora of enzymes for the breakdown of the macromolecules. The pancreatic exocrine gland contains acini and ducts. The pancreas contains this modified simple cuboidal epithelium called the acinar cells. These are then arranged in sac-like acini that are organized into 2 lobules. These acini cells produce and release the digestive enzymes. The secretions of the acini cells and the cells that line the pancreatic ducts form pancreatic juice, which is an alkalinic fluid that contains a lot of bicarbonate, or HCO3-, and this helps neutralize any of the acid. You also have these islet of Langerhans cells. These are the endocrine cells that contain the alpha and beta cells. These ones will secrete insulin and glucagon that regulate sugar levels and satiety. The pancreas secretes the digestive enzymes and bicarbonate. So let's take a look at the bicarbonate secretion. So at the bottom you have the capillary bed. On the top you have the lumen of the pancreas or the northern intestine. So over here, CO2 diffuses across a cell, combines with water, and with the help of an enzyme called carbonic anhydrase, gets converted into bicarbonate. The protons are going to be pumped out for an exchange of sodium, and that sodium gets pumped back out for potassium. It allows a buildup of ions inside the cell. Because there's more sodium on the capillary side, sodium will flow through and diffuse between the cell. Within the apical side, the bicarbonate can pass through because of the repelling of the negative charge of chloride. So chloride goes in, and then bicarbonate goes out. So now bicarbonate can bind to the protons and then create more CO2 to neutralize any acidity. Some of the key digestive enzymes include endopeptidases, which include trypsin and chymotrypsin. Their main function is to break down most of the protein. They're synthesized as non-active enzymes called zymogens, so they require the activity of an enterokinase to turn it on. So it activates the trypsin, which will then in turn activate chymotrypsin. There are also exopeptidases, such as carboxypeptidases, that will cleave amino acids down and break the peptide bonds. Lipases work with the bile salts to break down fats. Pancreatic amylase will hydrolyze the bigger chain starch to maltose, and other kind of nucleases, like deoxyribonuclease, ribonucleases, gelatinases, elastases.